Well, good morning, everyone. It's an enormous honor for me to give this Stanley Fahn lecture. And it's a pity that Stan himself cannot be here. He's still working on his recovery. And on this slide, I put on the Liberty Bell that I photographed when I was here, as I think freedom is a very high value these days. These are my disclosures. And what I would like to talk about today is a bit how it all began and where we stand now and where we are going to. And for me, I can reassure you it didn't start in '66, my research, but in '66, Professor George Bruin, who's at the bottom, he found this family with excessive startling. And he studied them by making videos. At the back you see his shotgun, and then he has this slow motion taking of the video with a clock in the front, and based on that they draw the startle reflex in these patients. So he was the first to describe this with a name. The disorder had been described once before. Let me see. I'm stuck already. Can you please? Yes. And what's hyperaplexia? Some of you might know, but others not. And it was the topic of my thesis that I worked on from '92 to '97 in the University of Leiden. And there are actually three main things. One is a stiffness at birth. And here you see a baby with hyperaplexia. And as you see, she's hardly moving, but she's a happy baby. That's me, pregnant of my first child. And when I lift her up in a second, as you can see, she is stiff. And when you turn her to the side, she remains as stiff as a stick. And that's very typical of hyperaplexia, a happy baby, but as stiff as a stick. And on the next slide, you can see very short-lasting stiffness that is induced by startling. On the left, you see her when she is about 1 year old, and you see gradually the stiffness disappears. And here you see this very short-lasting stiffness that is induced by sensory stimulus. And when patients exhibit this during their walking, they fall as stiff as a stick to the ground forward on their cheek or on their forehead without being able to stretch out their arms. And when you have a patient like that in the outpatient clinic, the only thing you can see is this exaggerated head reflection reflex. You tip them on the nose, and they have this backward movement of the head, and that's sort of considered a hallmark of hyperaplexia. And it's important to recognize this disorder as it's usually taken for epilepsy because clonazepam is very effective in preventing this stiffness. Can you please? Yeah. And what we did those days, when I came across this family and the film was given to me by Professor Brown, we decided to measure their startle reflexes by using polymorphy. And that's what you're seeing here. On the left is a control, and on the right is hyperaplexia. And this was my supervisor, Gert van Dijk, who helped me a lot with my thesis. And as you can see, there is a definitely exaggerated motor startle reflex, but the pattern is normal. So it's a normal reflex, but exaggerated. And then we had the chance to really look up also the clinical details of this large family. And what we noticed that there was what we call a major form, which is excessive startle reflexes with stiffness with startle and stiffness at birth. But there was also a minor form with just excessive startle reflexes. And what became clear in the pedigree is that once you had the minor form, the major form didn't reoccur, so it sort of stopped then. And at the time that I was doing that, and then it was still only Mendelian genetics, which is quite old-fashioned these days, then the gene was found. It appeared to be a mutation in the alpha-1 subunit of the glycine receptor, and that it's an inhibitory neurotransmitter in the brainstem and in the spinal cord, and then you can imagine this excessive startle and the stiffness. And we examined this Dutch big family, and it came out that only the patients with a major form of hyperaplexia had a mutation in this gene and not the one with the minor form. So that was very interesting. So we decided to measure their startle reflexes as well. And on the right, you see they have excessive startle reflexes, but on the left, you can see that the pattern is different. The latency of the EMG responses is much longer than the ones seen in the major form of hyperplexia. So we concluded that the major and minor form of hyperplexia are different, both clinically, but also genetically, and the startle response pattern is different. So we actually thought this might be like a learned behavior in a family where excessive startling is really a big issue. And we could confirm that genetic results in a cohort from Peter Brown, where I spent 1 year with in Green Square, that we had similar results in that cohort. And what's interesting, even if you do very rare disorders, there is also always a broader perspective. So we were asked to write a review on startle syndromes, and that was for us the chance to make a new classification. So the first one is hyperaplexia, as I just described to you, with the excessive startle, the stiffness, and stiffness at birth. Then you have clinically the stimulus-induced disorders. That means like startle-induced epilepsy or startle-induced PKD, because some of the patients with paroxysmal kinesogenic dyskinesias also have startle-induced attacks. And the third one is the neuropsychiatric disorders, where you have not, next to the startling, more behavioral features. And that was about the time I started in Amsterdam with Hans Speelman. And because we have an old relation with Indonesia, we had the chance to go to Indonesia and study one of the neuropsychiatric disorders, and especially LATA. And that comes from Indonesia and Malaysia. And we work together with Indonesian colleagues in that area. And I have always been supported by these disorders by Andrew Lees, as he's also very interested in these cultural-bound disorders. And what do these patients have? Well, they have this also non-habituating excessive startle response, but they also have behavioral features like vocalization, echolalia, echopraxia, forced obedience, and there is also a higher frequency of anxiety and depression. And just to show you how LATA looks like, oh, you have to put the sound down, please. They keep being startling. They also echo the stimulus that is given to them. And they also copy each other. But this looks very different from hyperaplexia. And we had the chance to also look at their startle reflexes. We took our machines with us to Indonesia. And on this picture, you see what we actually saw in the previous study in hyperaplexia, where you have this enhanced motor response that's originating from the lower brainstem. But what we found in LATA was much later, or not much later, but it was later. So the original response is like between 20 and 120 milliseconds. But in LATA, we found this secondary phase be much more enlarged. And that's in the old literature named the orienting response, or what is it response, and it's very variable, complex behavior and influenced by psychological factors. It's sort of like you're startling and you're trying to focus where the startling stimulus comes from. So that's actually really something else. So that, as it was assumed in the literature that it's similar to hyperplexia, it appeared to be very different. And just a short jump into the future here. If you look at this LATA, when I hear about these TikTok tics that are currently, that have been occurred in the pandemic, that looks a lot like LATA. They are not really tics, they are typical tics, and they have all these associated features that really resemble the LATA features, and they also occur in a closed circuit on the internet, as LATA is in this very certain area in the world. And I would like to mention this 2015 picture, as that was the first picture taken of a group of leadership, of female leadership in the MDS. Now I would like to switch, because I saw this article in Science, and budding scientists inherit career success or the lack of it from their mentors. And now I'm allowed to give this lecture. I consider this as a success, as you can imagine. So I would really love to give this success to my PhDs and fellows. And then there was a very nice quote in a paper from Anna Sotnica and Mark Edward, and they said, a neurologist training in movement disorder develops the skills of identifying and correctly subclassifying phenotype through experience and supervision by specialists in movement disorders. So I thought I love phenotyping, combining the clinical features with support of electrophysiology, the use of genetics, and then figure out what's going on, as I illustrated you in the previous slides. But then there is this thing, what is phenotyping? Is it just the clinical phenomenology? Or does it also include the electrophysiology, the genomics, metabolomics, radionomics? Probably yes. And these days, some people tend to say we should classify based on the genetics. And I don't like that. As being a clinician and a movement disorder specialist, I like to phenotype. And then there came this very nice paper, and there's going to be a lecture on it in the afternoon at 3 o'clock. And Mark and Anna, they describe here that if you look at the phenotypes, there are only, phenomenology, there is just a limited number of phenotypes. So the sensory motor system just breaks down in a very limited number of ways. So if we want to find out what's going on beyond those phenotypes, we have to find the circuits that underlie there. And those are the ways at the moment we interfere with by treatment, like with DBS. We are using this concept. And so they say we should go for the clinical phenotype because that's the start of our research, and that's very close to my heart. But there are a few things with clinical phenotyping that can be a problem. One is that there are overlapping features. As you all know, when you have a patient with chorea, you can have like jerky movements in there that really resemble myoclonus. And if you have dystonia, there can be tremor in there. So there are these overlapping features that might make it difficult to really say what kind of single phenotype is there. Furthermore, most of the patients not have just one type of movement disorder, but most of them have combinations that even make it more difficult to classify them. And it's not as a surprise, the experts usually not always agree that well. So there comes the problem, what is the right phenotype? And we could say, well, we are the mentors, so we know, and so that's the final word on it. But I don't think that's the way to go. We should discuss and look, and what are we seeing, and why are we saying that this is dystonia or myoclonus? And these three items make that it's a problem for, like I said, the diagnosis, the phenotype, the treatment, but also the evaluation of treatment, because if you treat something, you want to know what you're treating and where it is effective on. So what we actually want is that this expert opinion that's very subjective, it would be amazing if it could be a quantification and more objective. And there comes machine learning in, because they might help that might help us to improve our knowledge. So we decided to set up a story, and then you first have to realize, how does the machine learn? Because I think similar to databases, rubbish in is rubbish out. So you must put in patients that you're really sure of, that the phenotype is what you think it is, because that's at the moment the best we have. So we decided to go for the hyperkinetic movement disorders. As you might all know, that's where I'm working on. So we focused on tremor, dystonia, myoclonus, and myoclonus dystonia with and without a genetic mutation. And we asked many of you to serve as a gold standard. So we asked 3 experts, and there must be a very high level of agreement before we said, okay, this is this single phenotype. And then we had a very standardized setup with EMG, motion sensors, and 3D video cameras. They performed 21 tasks, and there were 16 sensors. This was this IMU together, so the EMG together with the accelerometry. Then we did the machine learning. Well, it sounds like we all did it, but I'm going to show you that we just started. And then from that, we will find clusters of patients, and that will be more objective. And what we started with is patients with essential tremor and cortical myoclonus. And some of you might say, well, that's easy going. You can easily discriminate that. I would like to show you this video. This is an essential tremor patient, and this is a patient with cortical tremor. And if you're experienced, you do see that it's different, but you can imagine if you're not that experienced, that it's actually quite difficult. We get a lot of referrals with patients who are thought to have essential tremor, but actually have cortical myoclonus. So we decided as a proof of concept to do the first analysis, and we took 19 patients with essential tremor, 19 with cortical myoclonus, and we decided of all the data we had to start with the accelerometry, as we know best about the analysis of the accelerometry. We did pre-processing, and we used the power spectrum, but you could also use like coherence or co-contraction or other features from electrophysiology, but we thought we start with the power spectrum. Then we applied the machine learning algorithm, which is the GMLVQ, and that's one of my last mentors, Michael Beal. He's a data scientist, and he's very experienced in this method. And what's nice about the method, as I will show you in a minute, is that it gives you back why. Why is it discriminative? And then we will have the results. So this is quite familiar to the ones in the audience who use electrophysiology. At the bottom you have the accelerometry measurements, on the top the essential tremor, and on the bottom the cortical myoclonus. And as you can see, it is different. And then at the top you see the power spectrum. So the orange one is the cortical myoclonus, and the essential tremor, you see this clear peak at 5 Hertz that we all know about essential tremor. Then we applied the machine learning, and as you can see, it was amazingly good. On the bottom left, the ROC curve shows that it's almost 100% discriminative when you use the power spectrum. And the 2D projection just shows you the prototype, the stars, and then all the other data points from the other patients. And as you can see, it's very discriminative. And then on the right, you have the relevance metrics, which is like a multivariate analysis of the power spectrum. And I show you this as this is the same as the relevant matrix on the left. And what you see there, the orange one is still the cortical myoclonus, and the blue one the essential tremor. And then on the x-axis is the frequency. And the higher up, the more relevant the frequency is in combination with other frequencies. And as you can see, again, this 5 Hertz, but what also shows is the steepening of in the essential tremor is also very relevant, because in myoclonus you have this broadening of the spectrum, and then if you have that, that's very discriminative between essential tremor and cortical myoclonus. And what's nice about this, it reflects a lot the power spectrum as you see above. So that gives you the feeling as a clinician that what's happening here is really what we are trying to look for, so that the system works. And it's not a black box, because I think we are always worried about this machine learning, that you just put everything in, there comes something out, and what happened in between. But here you can it's given back to you why. So if we come back of how does the machine learn, As I showed you, we have the 3 modalities, and then the machine learning, we create new groups, and then with the visual analytics means that you make a system that you give back the information to the doctors in a way that they understand. So this system is not like you just put a patient in and let's see what happens. It's meant to be that you have a patient and you think, could this be cortical myoclonus? You put the patient in the analysis, and then the system says, yes, for 90%, this fits the cortical myoclonus, and that can be given back to the doctor, and also the reason why you think it is, the machine thinks it's cortical myoclonus. So that can be very useful in the clinic. But what can we learn from the machine? And I think there is quite a lot there, and I think that's extremely interesting, because we create, based on the machine learning, when we have done more analysis, we create new groups of patients, and there will be essential overlapping and discriminating features if you see if they are the same as was meant by the experts. So that will give us a lot of information about what we actually see. And another thing is, are we creating new groups that we're not realizing when we put the experts in? And it would be amazing if we could develop new biomarkers. I'm really thrilled to wait for the analysis of the videos and the dystonia, because that will really be something if we could find something there that shows us why it is dystonia, because we know it's dystonia. But if I have to explain a fellow why it is dystonia, we have these definitions, but there are always also exceptions, right? So it would be very nice if we learn more about new biomarkers there. And also the use of clinical neurophysiology, because the data I showed you was actually 1 task and 2 sensors, so that means less than 1 minute of measuring was able to discriminate very well these 2 types of movement disorders. And I must be clear about, we only focus on the arms, so we didn't look at the gait or at the neck, but just on the arms. But it might really help in the use of clinical neurophysiology. And then what's going to be the future? Well, I told you about what the EMG and the motion sensors and the video camera, but we have a lot of clinical information as well. We have the genetics from the myoclonic dystonia. There are patients with and without genetics. What's going to be the influence of that? And we also have fMRI and PET scans of all these patients. So it's going to be an adventure with all these data. And I think for that we have to embrace the machine learning, because it's going to be very tough if we have to do that ourselves. So I hope I was able to illustrate you that although we're still doing the same things with video and clinical data, EMG, and coming over time the genetics, with the machine learning we will be able to integrate a lot of that, and it will help us to move forward about phenotyping. But I don't think it will replace it, because at the end we are the experts, and like the paper from Mark Edwards, it's very important that that's our starting point. So let's learn the next generation a new type of phenotyping, and I think that's really nice for the future. So a thank you for my mentors, if you've seen them on the slides going on over time. But also a big thank you to the leadership of the MDS that I've become friends with and who learned me a lot over time. And all my international collaborators from the functional movement disorder, from the dystonia, the electrophysiology, myoclonus, rare movement disorders. And a thank you to my Dutch colleagues who were involved in these projects as well. And a big thank to my group in Groningen from the Movement Disorder Expertise Center. And I think the major big thank you is to all the fellows and PhD students that I had the privilege to work with on all these projects. And I don't think we need machine learning to see, if we look at the left and at the right, that diversity improved a lot over time. And I think that's a great thing to do. And I really hope that we have this very diverse group of young people bringing us forward in the future. And all these people together feel like my movement disorder family, but I must be honest, there is one family that's more important to me. And I would like to thank you very much for your attention.